Hi everyone, welcome to Microbial Minutes. I hope you can all hear me. I haven't used this software in a little while, but I think I've activated everything. I'm on the line. If you can't hear me through your computer, please feel free to call in to the phone number that I provided. Sorry for the late notice, but we will next time be resuming the YouTube recordable one, and hopefully we'll be recording this as well to share with those who can't join us today. One other piece of business before we jump into what's the latest in the microbial sciences is that I realized in some of the emails and the meetings that I've been sharing that I may not have it been appropriately including the links to the Google Drive where a lot of the reports live if you want to read them yourself. So if you want access to the Google Drive and you haven't had it, please send me an email. I'm happy to share that with you. All of the papers that we discuss, as well as the meeting notes that have a lot of these links, are in there. So feel free to shoot me an email if those haven't been available to you. All right, great. Glad you guys can hear me. Okay, so what has been happening in the microbial sciences? We have 4 papers to discuss today, as well as a little bit about that cholera epidemic that I mentioned at the end of last time's session. There haven't been a lot of scientific reports about it, but it's been in a number of different places in the news, so I thought that it was something that would be important to discuss in one of these sessions. We're going to start with this topic, and the item that I chose to share about this was a commentary that just came out last week from Lori Garrett, who is well known for writing about infectious disease. Her commentary is titled "Cholera is Slaughtering Yemen, and We're Letting It Happen." And the take-home message from this is that cholera epidemic in Yemen continues to worsen and spread. And so for those of you not familiar with cholera, it is a waterborne disease caused by Vibrio cholerae. It's a bacterium, and it causes a famous watery diarrhea. It does this through an electrolyte imbalance that kind of tricks the epithelial cells into secreting a lot of their water contents, and this causes a very watery-like diarrhea, rice-like stool, it's also been referred to, and this is going to be important later on as we talk about this. And you may be familiar with an epidemic that was a little closer to home in Haiti that happened after the earthquake. That was found through molecular epidemiological techniques to be due to the United Nations peacekeepers, specifically some who traveled from Nepal, and were carrying that bacteria in their system, even though they didn't have fulminant disease. And right around the time that Hurricane Matthew occurred last year in October 2016 is when the crisis in Yemen seemed to be on the upswing as well. There was rumor of some of these cases, and this is tied pretty tightly, its spread is tied pretty tightly to the ongoing civil war in Yemen. And so back in October 2016, there was just rumor of some cases, and now as of July 2017, there are over 350,000 reported cases with over 500,000 expected reported cases by the end of the month. And keep in mind that reported cases are always underrepresentative of the true scope of an epidemic because there are many people who may be suffering at home who may not have access to become a reported case in a hospital or healthcare setting. So far, there have been nearly 2,000 deaths. Both the caseload and the number of deaths are increasing at 4 or nearly 4% per day. This is a lower growth rate than previously in the epidemic, but does seem to be on the upswing again. And again, this is tied very tightly to the civil war, and the results of this civil war going on in Yemen is that 14.5 million Yemenis of a total population of 25.4 million, so this is over half of the population, has no access to clean water, in part because of the bombing campaigns which have completely obliterated what little sewage system and infrastructure they had. This is still a developing country, but they did have a water transportation system and certain methods to clean some of their water before it was sent out to the homes, and this has been demolished by the bombing campaigns that have been ongoing. Importantly, cholera and the bacteria that causes it is not something that respects national borders, and cases have started to be reported in nearby countries in the area. South Sudan, Ethiopia, Kenya, and Somalia have all reported cases of cholera itself, with other countries reporting watery diarrhea and not necessarily testing for the bacteria itself. And one of the things that this bodes poorly for is the upcoming Hajj, which is the annual trek of Muslim pilgrims to Mecca in Saudi Arabia. These pilgrims come from throughout the world, including from these countries that are being affected by the current outbreak. And it's possible that not only will this affect the large accumulation of people in Saudi Arabia, but could also then spread back out to the native countries of these different pilgrims. So people are quite worried. Public health officials and infrastructure officials are very worried about what the effects are going to be if this outbreak cannot be brought under control. So one excerpt that I just wanted to read to you from Garrett's piece is here. There is, of course, a vaccine for cholera, and the UN system geared up after the 4th of July to vaccinate 500,000 Yemenis in a heroic mass immunization campaign. The need was 20 million doses, but the world stockpile of cholera vaccine had been allowed to dwindle, and manufacturers, put off by low profit margins, stopped making the life-saving medicine. Vaccinators hoped to target meager supplies wisely and slow the epidemic spread. But on July 13th, the WHO canceled vaccine shipments. The Saudis had refused to guarantee a ceasefire, which would have allowed the UN safe passage and a few days window of immunization possibility. And so this was really unfortunate. So the ongoing civil war has two different factions backed. It's basically a war by proxy between an Iran-backed and a Saudi Arabian-backed faction, and they could not agree to stop fighting for the short period of time that would allow health officials to go in and provide this vaccine to those at most risk, at highest risk of coming down with the disease. So that was extremely unfortunate and is ongoing. So this is just happening a few weeks ago. There are ongoing talks to try and provide some sort of sanctuary or brief halt in the fighting so that this vaccine can be provided to the populace. This was a very widely shared article, I think, in part because of the harsh tone that Garrett took, saying that this is something that both the military and those who might, the United Nations and other people who might be able to influence some of this talk should be taking a stronger stand on. This has been covered in Science. They covered it with a newspaper or a journal article titled "Cholera Vaccination Campaign in Yemen is Dropped." Again, this is front of the magazine type of stuff. There haven't been a lot of epidemiological studies or other types of scientific reports, although I expect those will be coming up in the future months. So this has also been broadly shared on social media. One user quoting Garrett by saying, "Yemen has the worst cholera epidemic in modern history. Even the new Director General of the WHO plays the name game," writes Lori Garrett. And this name game is the idea of calling cholera this watery diarrhea. So there are some countries that have not yet reported cholera as cholera, but say that there is a local outbreak of watery diarrhea. And this is in part because it's important for countries in a global society to be able to interact and not have things such as infectious disease outbreaks that might impede business or might stop travelers or tourists from coming to their country. And so finding the true scope of the outbreak is important, and it's important for public health officials of countries to honestly claim if they are having outbreaks of cholera. Another user here says that key messages from Lori Garrett include our failure to intervene, avoidance of naming it cholera, and collapse of cholera vaccine supply, and more. So this is clearly an ongoing story, and we will update occasionally on this Microbial Minutes. This is one of the largest outbreaks of cholera that has ever been recorded. I mean, they haven't all been recorded, but this is truly something that will be affected by both global politics and public health measures. All right, moving on into our research articles. This is a article from PLOS Pathogens entitled Streptococcus galliticus subspecies galliticus promotes colorectal cancer tumor development. And the take-home message is that bacteria directly promote colorectal cancer. And this is an important finding because Streptococcus galliticus, which the authors refer to as SG, has been associated with colorectal cancer for a number of years, often found in conjunction with tumors and specifically tumorigenic areas of the colon. But association does not always mean causation, and they wanted to ask the question of whether SG plays a role in causing tumor development, or whether the SG is simply taking advantage of a niche in which it grows well. It could be that the tumor provides extra nutrients or some place that is somehow beneficial to the SG in order to grow there. And so they wanted to look at this in a more systematic way. And so one of the things that they did was to take tumor cell lines and grow them in the presence of this SG and other related and unrelated species. And you can see in this, hold on just a second. Look, I've been using the wrong arrow. Forgot about this part here. So you can see here there are 3 different SG strains that they tested that promoted larger, more cell growth relative to untreated cells. So here's the cell number, and you can see there is more cell numbers of these tumor cell lines when they are grown in the presence of SG than in the presence of other Streptococcus species or in other unrelated but also commensal species such as E. coli or Lactobacillus lactis. They also found that this promotion, this tumor promotion, is associated with a certain phase of growth. So it's not actively growing cells that promote tumorigenesis, but those that have reached stationary phase or are not actively dividing, yet still metabolically active. And they did not have an explanation for why this is, but they said that it is not necessarily like a newly colonizing SG that is going to be promoting this tumor cell growth. They further confirmed this by looking in a mouse model of colorectal cancer. They added a carcinogen that is known to cause tumors in the mouse colon, and then colonized the mice either with SG, with another colonizing bacterium, Lactobacillus lactis, or with no bacteria. And you can see both here the tumor burden, which means the number of metastases or the number of tumors within that mouse colon was higher, as well as the degree of pathology. So the dysplasia is one measure of how severe those tumors are, and you can see that there were more tumors and they were more severe when the SG was present, confirming that there is an active role of SG in promoting tumorigenesis, but that it is not the sole reason. So you can see that there are also tumors within the other test groups as well. They also looked at some human samples, some patient data, and found that there was an association of people with tumorigenic tumor samples relative to those that had just normal colon samples that were tested, looking at the presence of this SG. So again, showing that this is something that translates into human disease, which is always important when looking at an animal, a mouse model. Yes, I hear the static too. So I see that there's some static for those of you who are listening on the headphones. I hear the static too. That's probably because not everyone's muting their phone. So if you guys could mute your phone, then that would probably eliminate that problem. Thanks for the note, Catherine. So this story was picked up by Science News. It was shared broadly because this is yet another microbe that is associated with a type of cancer. Now, it's not solely responsible for cancer. As you saw, some of the mice that were not colonized with SG also developed tumors, and some of those were also severe, but it may play a role in promoting tumor growth. And so in this particular study, in the Science News story, they said that the author of this story said that further investigation could help researchers find ways to block the microbe's action, leading to better treatments for colorectal cancer. And so that's really one of the major findings here, is that if for example, an antibiotic or something that could inhibit whatever it is that stationary phase bacteria is secreting that helps to promote tumors, can also be targeted in addition to any type of chemotherapy that targets the tumor cells. That is something that might help patient outcome in the long run. All right, moving forward, we have a very exciting story that may have crossed into some of the mainstream media. This one was probably most broadly shared of all of the um, scientific reports that we're talking about. This is titled CRISPR-Cas encoding of a digital movie into the genomes of a population of living bacteria. And the take-home message from this is movie storage and E. coli uses DNA to store data. Now, DNA as a data storage mechanism is not a new idea. A lot of research labs are looking at this, including George Church's lab. And as you can see, George Church is the senior scientist here. DNA is a repetitive polymer, so kind of like computer codes that are made of ones and zeros, you can imagine that it is repeating subunits of A's, T's, C's, and G's instead of ones and zeros. So there's a little bit, there are more characters, but it is something that is basically composed of these four different subunits, and it's also a very stable molecule. So you can find and sequence DNA from old mummified mammoths or Neanderthals, and so this really speaks to the longevity of DNA as a way to store data. Additionally, it's very small, so we have in all of our human cells about 6 feet worth of DNA that is wrapped up into the nucleus of our cells. So there are lots of different characteristics of DNA that make it a potential storage molecule, and that's what this lab was working on, was to actually use it as a way to store information. And they were looking at images because images require a lot of storage space, right? So as our smartphones have gotten better, it's only more recently that we're able to take a lot of photos, and at least in my case, I am always running out of space because I take really high quality photos. And so you could imagine if you could just upload that photo onto a little bacterium, you'd be able to clear space on your phone, for example. And so they were asking the question of whether DNA could be used to store image data. And so they did this by creating DNA snippets that correspond to pixels in an image. And they used a code that was different than the code that is used for turning DNA into amino acids, but with a similar idea of 3 bases being a piece of information. And each snippet of DNA had one area that gave the pixel position, and others that gave the shading of what color what shade of gray, I should say, that snippet would be. And so they did this in 2 different images. One was a still image of a hand, and one was actually a moving image. And so in this moving image, they were able to get 5 different frames encoded into the bacterium. Each frame was 104 DNA fragments, each of those being 33 base pairs long. So that's basically 104 pixels. So it's quite pixelated. You can see here it's not the same quality as our iPhones just yet, but you can get an idea of what this particular image is. This is a famous movie that was used in the late 1800s in order to demonstrate that horses actually do pick up all 4 hooves on all 4 of their feet when they're galloping. And they put 5 of the frames of this movie into the E. coli, and it took them 5 days by cloning 1 frame per day into the E. coli subsequently. And then they had to figure out how to get the information out. So once you put the information in, cool, you've got it in the E. coli, you can grow it, you can propagate it, you can share it, you can freeze it, you can do whatever you want with that DNA within the E. coli. Then you have to extract it. And so they sequenced different numbers of reads, which would give them different coverage and allow them to extract different amounts of that information. So as you can see here, with 5,000 reads, there's still a lot of missing information. And so they increased the number of reads several orders of magnitude, but they saw diminishing returns. So after 1.3 million reads, going from that to 4.3 million reads, there was a little bit more clarity, and they were able to fill in a few of these missing pixels that you see here. But they found that there was diminishing returns, and they determined that 1.3 million reads was sufficient in order to extract that information and then turn it back into a movie. And so this was a really exciting finding. There was a commentary that accompanied this Nature piece, and I sent that out as well. From the commentary, in the commentary they spoke to first author Seth Shipman, And I found his background very interesting. He's a synthetic biologist at Harvard Medical School. And it says here that while studying brain development, Shipman became frustrated by the lack of technique to capture how cells in the brain take on distinct identities. This inspired him to explore the idea of making cellular recorders. So the idea here is that Shipman may eventually use some of these techniques in order to see how brain development occurs and how cells differentiate into the distinct types of neurons or other cells that they are. This is obviously very rudimentary. If you look at that pixelated image, it is not quite as refined as if we were just to use the technologies that we have available to us now. In that same commentary, there was another quote from synthetic biologist Victor Zhernov, who says that this is like the first airplane flown in 1903. It was just a curiosity, but 10 years from that, we had airplanes almost like what we have today. And it's true, once a concept is demonstrated, there's a confluence of researchers who will collaborate and even compete, and that will lead to the new technologies that we can apply in our everyday lives. So this is, I think this is really exciting and very promising. This is also written up in The Atlantic. Scientists can use CRISPR, which was CRISPR's a genome editing tool and was how they were able to add in those DNA snippets. Scientists can use CRISPR to store images and movies in bacteria. It's a step toward turning microbes into living hard drives, and that may very well be what the future holds from us in 10 years. So moving forward, we're going to talk about a second, since the new scanner will be a DNA sequencer. Yes, Stefano, it could very well be. And in fact, without the, even though CRISPR is the technology that allowed them to make this data storage thing, without the ability to sequence very cheaply, this won't come to fruition. But sequencing, the cost of sequencing is decreasing so much that you can get for $1,000 those MinIONs that plug into your computer, and then that can be used in order to upload directly into your computer. So it's possible that maybe someday you'll just have some sort of bacteria that you put into your hard drive or put into your S drive or something like that. All right, so moving on to the next synthetic biology paper. This is from Science Advances, and this is from Virginia Cornish's lab, entitled A Modular Yeast Biosensor for Low-Cost Point-of-Care Pathogen Detection, with the take-home message that an engineered yeast is a low-cost point-of-care dipstick tool. So we're talking again about synthetic biology, which takes well-characterized DNA pieces and then uses them to answer some sort of question. In this case, they are rearranging the input and output of a genetic circuit, and I'll show you what the genetic circuit is in the next slide. And then you can have this circuit answer particular questions. For example, if you want to know if there's arsenic in water, you can have an E. coli that detects arsenic, and then given that arsenic presence, it will activate a genetic program that causes it to glow green, like a GFP or something like that. And the problem that these researchers are addressing is a little more serious. It is that many regions in the world lack infrastructure for sophisticated diagnostics. So they had the idea that maybe we could make a genetic circuit that can tell us whether or not people are sick with particular microbes. And so to do that, they made a genetic circuit, and this is what the schematic of a genetic circuit looks like. So here, all of these different filled in arrows are different genes that are controlled by specific promoters. The promoter is the region directly upstream of that gene that turns on or off the expression of that gene, whether or not that gene will turn into its protein product. And the culmination of this genetic circuit being turned on is that it activates lycopene production. Lycopene is, you may be familiar with it as I think it's an anti-carcinogen in tomatoes, but it's basically a pink pigment that is found naturally in plants. And the turning it on depends on what this yeast sensor is. So they have a number of different sensors that they incorporated into the genetic circuit, and these were specific for different pathogen molecules. And what they were testing were different pathogenic fungi. So they were using one fungus, which is the yeast Saccharomyces cerevisiae, same as baker's or brewer's yeast. And they were using different pathogens in order to test and make a proof of principle type of input to get the output, which is the color change. And while it shows here a yeast sensor, it's specific for whatever pathogen they're detecting. They cannot use one yeast strain or one genetic circuit that's going to detect all different types of fungal pathogens. They had to make one. In this case, they had a purple fungus, a green fungus, and a pink fungus, and they'll require their own specific receptors that are then encoded into this genetic circuit. And then you can have a panel of different Saccharomyces cerevisiae, and only the panel that changes color is indicative of which of those particular pathogenic fungi are present. They tested this against Candida albicans, which is a closely related or a somewhat related fungus to Saccharomyces cerevisiae and is found as a commensal on many people. It's an opportunistic pathogen. They tested this against a primary pathogen, one that is only found in the context of disease in humans, Paracoccidioides braziliensis. And you can see here in panel B what it looks like when they run this assay. So here are two Saccharomyces cerevisiae samples. I don't know what the scale is, but I think that's something around the size of a pinky finger. So this is definitely macroscopic. You don't need any fancy equipment in order to find out whether the sample is going to change color. And then after about 12 hours of exposure, you can see here that whatever they are testing for changes in the positive control, and in the negative control, there is not the same amount of color change. And so if you were running a whole panel, you would expect most of them to remain colorless like this, and then whatever pathogenic fungus is present, you would be able to say, oh, there's your positive. So that's what disease this individual has, or what microbe is currently infecting this individual. And so this is really great, not only because of the ability to work as a diagnostic tool, but the application is extremely cheap. It's easy to transport. You can dry down baker's yeast, right? If you've ever made bread at home, you can just dry it down, and then you have to re-wet it in order to activate it again. You can do the same with this type of diagnostic technology. And so this was also written up in the Atlantic, turning baker's yeast into a disease sensor. You'll notice both of these articles are written by Ed Yong, who was at Microbe, the individual who was interviewing the astronaut, Kate Rubins. And in here, in this article, Virginia Cornish is interviewed, and she said that our vision is that we can give the yeast to a resource-poor country, and they can grow the biosensor themselves, just like they can brew beer. And it's true, this is a self-propagating type of diagnostic where you can make more simply by diluting into fresh medium, give them some simple sugars, the yeast will continue to grow, and then there was another panel that I'm not showing you, but they had to test it against a number of different types of patient samples. So they tested against serum, blood, saliva, urine, to show that this can work against a wide variety of different types of patient samples. This was also written up in Discover magazine. Links are in the slides, which I said I'm happy to email you if you don't have access. And this was also very widely shared on social media, with one user here saying this is a really cool idea, using native STE2 receptors and modular yeast biosensor for sensitive pathogen detection. So really so Catherine's asking, how stable are the strains and how quickly do they acquire mutations that would give false negatives? I am not sure. I don't know that they've tested that. This was really a proof of principle type of experimental setup. I'm sure that that's something that would be important to maintain, and maybe there would be some sort of positive control that you would include with that, or you could send it out for sequencing every once in a while. It's true you don't want too much activation, but the scientists did have trouble not with overactivation of lycopene production, but actually under activation. They had a hard time getting enough color that you could visualize it without using some sort of sensor, so they had to add in a cofactor that was expressed at the same time. So I think if anything, you'd get false negative. False negatives. That's what you have. So it's it's possible, and that's something that they'd have to be careful of going forward. A lot of the write-ups on this did mention cholera, which I found very interesting given our previous discussion about cholera, but none of the research was actually done. It was all done with these different pathogenic fungi. All right, we are closing in on a half hour, and I'm really trying to keep these to 30 minutes. So I'm going to go through this very quickly. This last paper that I found on the day that I was sending out papers. This was making a lot of waves on social media. I thought we've got to include this in Microbial Minutes. This is yet another Nature paper. They've been really publishing a lot of cool microbiology lately, titled Rapid Elicitation of Broadly Neutralizing Antibodies to HIV by Immunization in Cows. The take-home message is that immunized cows produce broadly neutralizing antibodies to HIV, and these broadly neutralizing antibodies and development of these has been one of the goals of HIV vaccination studies in general. So before we move forward, I just want to very briefly talk about what that means. What is a broadly neutralizing antibody? Antibodies do all kinds of things within the body. They can do cell-free viral neutralization, where they bind to a virus and block its interaction with a target cell, so the target cell cannot be infected. They can inhibit cell-to-cell viral spread very similarly by binding and interacting with that virus itself or with parts of the infected cell that are necessary to release the virus. They can activate other immune cells. So there are portions of the antibody that might recognize an infected cell and then activate another type of white blood cell that could then come and kill the infected cell and keep it from spreading new progeny viruses. But this viral neutralization, this is really key for a good vaccine to produce neutralizing antibodies so you don't get that initial infection, especially with something like HIV, where once it's inside of its target cell, it's going to incorporate into the host genome. And broadly, so that's what neutralizing antibodies mean, and broadly neutralizing means that it needs to neutralize across a wide variety of different viral strains. And HIV mutates quite a bit, so there's a lot of different strains out there. And it mutates a lot within its glycoprotein called ONV. And this is a schematic here of HIV, the virus. And here is the glycoprotein, which is important to interact with the host T cell when it infects the CD4 T cells. And a lot of these broadly neutralizing antibodies that have so far been somewhat promising in studies are to the conserved regions of ONV, which tend to be, so here we are looking at a close-up of that glycoprotein, which are at this lower part that is right next to the viral membrane and very hard for antibodies to access. They're hindered by their size from reaching that area of the protein. And so one thing that I did not know in reading the background here is that cow antibodies are bigger. They have a different structure, slightly different structure, and they can reach a larger epitope than human antibodies can. And so the researchers here wanted to see if immunized cows could generate broadly neutralizing antibodies. And so they ran this in a few different experiments. The setup is shown here on the left. They immunized some animals and boosted them twice. And the second experiment is shown in blue, where they immunized animals and then boosted or exposed them to that antigen again for 5 times. And then they looked at the ability of their antibodies, of the antibodies collected, to neutralize or no longer infect, allow viruses to infect cells. And they do this by looking at, so there's lots of different antibodies that are produced, and they would take a single antibody producing cell and clone or expand, look at one single antibody from an array of a lot of antibodies that are being produced by that animal. And so here in this example, here's one animal, and here's all the antibodies that they collected from that animal here that are labeled basically by what position they are on the plate when they're isolated. And then to test how well they neutralize, they look at how many times they can dilute it. So the more times they can dilute it, the better it is at neutralizing that virus. So some do not neutralize at all. You can see 10 to the 0 here, so they do not neutralize. You cannot dilute them. And then some of them neutralize okay. You can dilute them maybe 100-fold. And one of them over here in this particular example of this cow neutralizes almost, you can dilute it almost 1,000-fold and still see that neutralization effect. And you can see that in the second cow here, they were slightly more effective, and that when they had more boosts of that antigen, they had more neutralizing antibodies, or more neutralizing to a greater capacity. They were able to dilute them further. However, in general, most of the antibodies that they found here are able to neutralize to some extent, even if they can only be diluted maybe 1.5-fold or something like that. And so this was very promising. And they do not, the scientists were surprised at how many of the antibodies that they collected from the cows were able to neutralize this, this particular HIV. And they tested against a wide variety of different HIV strains to show that it's broadly neutralizing, not just against this one strain here. And so this was very promising, and it's suggestive that maybe testing in animals that have not evolved with this pathogen, like cows are generally not exposed to HIV, might be able to tell us something about how their immune system reacts in a different way than human immune systems. So Stefano is asking, do I know why the experiment was done in cows? And it's because of the difference in their antibodies. Cows have different structures of antibodies that allow them to cover a broader epitope, which I was unaware of until I read the background of this paper. And so are humans similar to cows? Humans are similar to cows in that they make antibodies, but their antibodies cover the region that the antibody binds to is smaller in people than it is in cows. So that's what they thought, that maybe it could reach a broader region. But the fact that they found so many neutralizing antibodies, so they expected to be able to find some neutralizing antibodies, but the fact that they found so many means that there might be a difference in the way that the cows react to HIV as well, that they will follow up. And in fact, the first author, Devin Sok, was quoted in Stat News as saying it was an alignment of the stars where we had veterinarians, cow antibody scientists, and HIV scientists all talking and came up with this relatively simple question to test. So this is a demonstration of how important it is for people in different fields to talk to each other and understand the science going on in other fields, since I did not even know that cow antibody scientists was a field, a very specific field, but one could imagine in agriculture that it's important to be able to vaccinate herds of cattle, for example. So of course there are cow antibody scientists, but how often do cow antibody scientists talk with HIV experts? Probably not very often, and it's only in discussing these various fields that we were able to come up with a convergence where you might be able to come to identify a new hypothesis or a new approach. So this was written up in a number of different places. Stat News is linked here. The Scientist, HuffPo, Time covered this. And in Time, they interviewed NIH Director Anthony Fauci, who said the kind of understanding we get from studying this is an understanding of the mechanisms whereby the cow's immune system is capable of creating these antibodies. And so that's something that will be followed up, I'm certain, in future studies. So thanks for sticking around for a little extra. I am trying to keep these to around 30 minutes, but I'm always happy to have some interaction. So this week, we have discussed several things going on that are important in the microbial sciences. We talked about the cholera epidemic in Yemen that continues to worsen and spread, bacteria that are directly responsible for promoting colorectal cancer, moody storage in E. coli, which uses DNA to store data, engineered yeast are a low-cost point-of-care dipstick tool, and immunized cows produce broadly neutralizing antibodies to HIV. So hope you guys have enjoyed. I believe this will be recorded, and if so, I'll be sharing the link if you want to share it with colleagues who couldn't join us at lunchtime. I am hoping to make these more broadly available for people who can't necessarily come at the designated time, just in case you want to find out what's going on. And with that, I will see you guys in another couple of weeks to discuss what's hot in microbial sciences. Have a good rest of your week, everyone.